All right. So in today's session, with respect to what we're talking about, the biomolecules, we're going to continue about learning about enzymes. Okay, it's going to be very interesting. So let's get back to where we stopped. Right. So in the last class, we were talking about metabolism, and we did not get to talk about enzymes. So let's start from here. So while we talked about metabolism and how organisms have to perform metabolism to stay alive or to show the living state. So one of the key aspects with respect to all these reactions that happens chemically should be attributed, or you have to give thanks to enzymes. Okay. So let's see how these both are linked. So when we talk about any of these metabolic processes, we understood that there is two things that is happening in our body. One is a constructive process, other is a destructive process, right? In constructive process, what we're doing, we are spending some energy to build something. And in destructive process, we are breaking down the complex things to gain energy, right? So these two things were happening. And to achieve that, some amount of energy have to be spent in all chemical reactions, correct, right? So now just imagine a body, and in this body, now for performing a very simple task, imagine you're applying all the energy. So if you're spending more the energy than finding the ways to conserve it, now what will happen? Your body have to find too much of food source or too much of energy source to keep this engine running. So to help us to balance this energy demands, enzymes come and play a very, very important role. So when we talk about enzymes, enzymes are, what are enzymes chemically? So enzymes are similar to that of your catalysts. So what are catalysts again? So for example, you might have studied in any of your chemical reactions in your chemistry class that whenever there is two reactions that is happening, one substrate is converted to a product. Now you put something like catalyst, which will enhance the rate of reaction. That means this, it will speed up the rate of reactions. Or for example, if any one of the chemical reaction is taking 1 hour of time, or it is taking too much of temperature, let's take that it is taking over 700 degrees Celsius, right? So this is without the application of catalyst. Now with the application of catalyst, let us say catalyst is helping us to reduce the reaction time in 15 minutes. I'm hitting 15 hours, okay, in 15 minutes, and the temperature requirement would be 90 degrees Celsius. So this is possible, right? With less energy, in the presence of catalyst, we are trying to get the product in a very short period of time. So this is what happens in all the inorganic chemical reactions that we talk about, right? Whereas when we talk about in a biological system where thousands of biochemical reactions are happening spontaneously, okay, so here also we require some catalyst kind of a substance to save us some energy and time. So those are nothing but enzymes, okay. So can we call like whatever the inorganic catalyst we use, can we do it in the biological system? Not really. Even enzymes have their specific property. Okay. So what are enzymes basically? By definition, we can just tell that enzymes are proteins. Correct, right? So the protein part is what the enzyme is made up of. So what kind of a protein is it? See, simple sequence of an amino acid. Like for example, if you take a polypeptide chain that we talked about, this polypeptide chain can represent an enzyme. So that means they can have both primary, secondary, and tertiary structures like that of your proteins. Understood? So these enzymes are basically called as ribozymes. Okay. So they also act like an enzyme. Okay. So they're basically proteins. Now, how do enzymes perform specifically? See, enzyme, what it does is it goes and binds to a specific region in the substrate, not product, substrate. So substrate is something in which the enzymes act upon. So when they react with the substrate, they go and bind to the specific regions on the substrate, and that will help in the transformation of this substrate to something else so that we can get the product in an early stage. So where does it bind? The site at which the enzyme binds. For example, we have an enzyme, right? So if an enzyme goes and binds to this region, that region where it binds is called as an active site. Understood? So as I told you before, proteins are structurally functional molecules, right? So their structure is very, very important for their function. So that is why what happens. So they go and recognize the specific regions on the substrate that can go and attach to it and bring about a chemical reaction. Understood? So these are called as active sites. Okay. So let us talk about a general chemical reactions to understand the activity of enzymes or how they perform their activities. So enzymatic activity is similar to any kind of any kind of chemical reaction, I can tell. So before we head on to that, I hope you understood till now what we're talking about enzyme. It's a very brief introduction about it. Enzymes are nothing but proteins, right? Like that of your catalysts. So we have in inorganic chemical reactions, we have catalysts that will save us energy and time and bring out of products very early. The same reaction does enzymes also do it, but in enzymes they are proteins. Okay, so they have primary, secondary, and tertiary structures like that of your proteins. Okay, so enzymes recognize a specific binding site to on the substrate. So this site where it goes and attached to the substrate is called as an active site. Okay, so if you talk about let's say that in inorganic reactions, right? So we talked about saving time and saving energy and catalyst playing a role. But what happens is, if you take the reaction happening every 10 degrees, what will happen, like higher the temperature you go, faster will be the reactions, right? Whenever you talk about any inorganic reactions. But in biochemical reactions, what happens is enzymes are very constrained to a temperature, specific temperature. So sometimes what will happen when the enzyme or is put in a environment where it is more than 40 degrees Celsius, many enzymes will degenerate. They will not function. Why? Because they're proteins, right? In proteins, you know that at higher temperature, proteins become denatured. Okay. Similarly, enzymes also lose their nature, so their structure will change so that they'll become ineffective. In lower temperature, what happens? The same enzymes, they freeze and they become dormant. They don't destroy their structure, but they will stop performing any activity. Understood? That is one of the key characteristics of an enzyme, being a temperature constraint. So anyways, let's talk about understanding enzymes through a chemical reaction. So enzymes perform a chemical reaction. So what kind of chemical reactions do occur in our environment? So when we talk about any reactions, it can happen in two ways. One is a physical change, other one is a chemical change. Correct, right? So there are two types of chemical changes that we can undergo. One is a physical one, other one is a chemical one. What happens in a physical change? See, in physical change, usually the matter gets rearranged. For example, you have your ice cube. When the ice cube changes its state to a liquid, so now what happened? The state of that particular matter will change, and here there is no breaking of bonds. Okay, the breaking of bonds do not happen. So a simple example: ice formula is also H2O, and then water formula is also H2O, and water vapor formula is also H2O. Here what happened? There is a change. Here only the matter state has changed. Apart from that, there is no breaking of bonds that is taking place in a physical change. Simple. So when we talk about chemical change, in chemical change, what happens? The bond breaking happens and then bond making happens. So take an example of an inorganic chemical reaction. So we have barium hydroxide, which is reacting with sulfuric acid. So we get barium sulfate and water. So in this reaction, what happened? First, the bond has to be broken down between the barium and the OH group, and then a bond has to be broken down between the hydrogen and the sulfate. Now what will happen? The bond making will happen, barium and sulfate, and then water. You would have studied more about this in chemistry classes, but yet to understand a basic chemical reaction, what is happening? A bond breaking happens and then bond making happens. In physical change, it doesn't happen. Correct, right? So for in both of this reaction, whether you take a physical change or a chemical change, what is happening? You get a product. Ice cube to water or water to water vapor is a product. Similarly, when you talk about chemical change, you start with something like this is your substrate and this is your product. Correct, right? Any chemical reaction that we are doing, our aim is to get the product. Simple. Whatever the product it might be, we don't want it. But all we're concerned about is if we perform something, we have to get a product. So as simple as that. So why product is very, very important for us? Now, product is important because we have to measure the time it takes to complete this particular reaction. Correct, right? So if I have a substrate and if I require some time to get a product, okay, so what does it give me? It gives me a rate of reaction. So, okay, I have to wait this long with this much amount of concentration to get these many products. So the product determines everything, like how much time it has taken. So when we have a rate of reaction, we can simply call it as the velocity of a reaction, because there is a direction to which your chemical reaction is happening. Correct, right? Either it goes from substrate, it also always goes from substrate to product. Correct, right? So it has a direction. One direction is there. That is why we call it as velocity. We don't call it as speed of reaction. So velocity of a reaction is determined, okay, by the time it takes to convert a substrate to a product. Correct, right? So there is a rule for every chemical reaction. What is that rule? Whenever we talk about the factors such as temperature, okay, if you change this temperature every 10 degrees Celsius, either you go higher 10 degrees Celsius or you go lower 10 degrees Celsius, what happens is either the rate doubles, okay, or it decreases by half. So what does it mean, students? So when we have a chemical reaction, in simple terms, it takes some time to finish that particular reaction to get a product. So that we call it as velocity of a reaction. So when we have velocity of reaction as our concern, okay, in a general chemical reaction, how does the velocity change? For example, I told you that in many inorganic reactions, temperature plays a very important role. Higher the temperature you go, the faster the chemical reaction happens. I told you in inorganic reactions. Similarly, okay, in all the chemical reactions, if you combine, see, just by taking temperature as a factor which influences a chemical reaction, for every 10 degree increase or decrease, what are we having? We are having a change in the direction of this chemical reaction, right? Either it doubles, so that is a change in direction, or it decreases, or that is also a change in direction of a chemical reaction. Correct, right? So this is happening in any other chemical reaction. So now, if you change your focus to the rate of reaction when it comes to enzymes, activity of an enzyme, okay? So when we take an enzyme, we know that enzymes are used in a chemical reaction to catalyze a specific reaction. Correct, right? Let us take an example. How does enzyme actually influences a chemical reaction? We have a regular chemical reaction that is happening in our body. So we know that we respire. I mean, like we breathe, right? When we breathe in, we try to do that because our tissues need oxygen. And the byproduct of the respiration is carbon dioxide and water. You know that, right? So when we break down this glucose, we have the end product is carbon dioxide. So carbon dioxide is not good for your tissues. Now what will happen? The carbon dioxide has to be removed from the tissues through your lungs. You have to breathe out carbon dioxide, right? So how does it happen? How does carbon dioxide get carried from the tissues all the way to your lungs? So that happens through a chemical reaction in your blood also. So in our body, in our blood, what happens is the carbon dioxide that is released by our tissues, okay, reacts with the water. So if this happens in a normal, okay, I can do this. I will take carbon dioxide and I'll use water and I'll try to get them reacting, okay, in a physical environment. For that, I'll be using catalyst, okay. So what happens? I get carbonic acid as a result. So this carbonic acid, if I have to get it without the help of an enzyme, okay, we have an enzyme called as carbonic anhydrase. Let us say that it is absent. So if I just use this reaction, I can get only 200 molecules of carbonic acid in every hour. This is so slow, correct, right? I don't have an enzyme. I don't have a catalyst. If I just perform this experiment where the carbon dioxide or water reacts to give carbonic acid, now it is taking an hour to give me 200 molecules of my carbonic acid. Whereas if I use an enzyme that is carbonic anhydrase, okay, the same reaction in our body it gives me around 6 lakh molecules of carbonic acid every second. Can you see the efficiency and speed of reaction that happens with the help of an enzyme? So this is a very brilliant example to understand how the enzyme can influence the speed of a reaction. Okay, are you guys understanding? Are you following what we're talking about? Yes. Students, are you understanding? All right. So let's go back to, okay, hold on. Nice. If you have any doubts, you ask me, okay? So we're talking about how enzyme can influence a reaction. Okay. Now, when we talk about another aspect of an enzyme, so let us say that we have a particular process in human beings, okay? So which is called as glycolysis. So does glycolysis happen even in bacteria? Yes, it happens. In yeast, does it happen? It happens. In many all organisms, glycolysis has to happen. Why glycolysis has to happen? Now here we have glucose as a very primary source of energy in many or in almost all the organisms. So this glucose molecule, our body has learned a way to break this glucose molecule into simpler forms to gain energy. So this is very natural. And glycolysis, usually what happens is it is a metabolic pathway where you have a 6 carbon atom glucose, which is broken down into 3 carbon atom and 3 carbon atom, which is called as pyruvic acid. So pyruvic acid, 3 carbon atom. Glucose has 6 carbon atoms. So when we break it down, it is split into half. Okay. So in this chemical reaction, that is where the glucose molecule is broken down into 2 pyruvate. Okay. So this is called as a glycolysis. Now look here. Whenever in this process is happening in our body during the respiration, where the glucose is broken down into pyruvate. Now what happens is, under we have two conditions. Okay, one is an aerobic condition, other one is an anaerobic condition. An aerobic condition is where, see, I'm very relaxed and my body is getting and tissues are getting sufficient oxygen, and my body need not take any extra risk. So all my tissues are getting enough oxygen. There is no much work. Whereas when I'm started running, so when I'm running, what will happen? There is a lot of work done. There is a lot of energy that is required. More of glucose is burnt, and all the oxygen molecules I'm putting in the body is used up for the process of breaking down this glucose. And that will lead to an anaerobic state where I don't have enough oxygen to give to my tissues, okay, for this process. So now what will happen? Whatever the pyruvate that was converted will start turning into a lactic acid. So this lactic acid accumulation happens in my tissues when I don't have enough oxygen, right? So when this lactic acid is accumulating, suddenly I get cramps, a sudden pain, where my muscles have contracted and they have stopped relaxing. So at this state, I'm facing tremendous amount of pain. So this cramp formation happened because of the lactic acid formation during this process of glycolysis. Correct, right? Now, if you take the same anaerobic condition in yeast, so if I take that, yeah, yeast also what happened, glycolysis will happen. But here, when yeast do not have enough amount of oxygen, it forms ethanol. Like for example, we use yeast in the production of wine, where we get ethanol as a byproduct. So we have ethanol formation in one hand, and we have lactic acid formation in the other hand, but we started from the same thing, that is glucose. Glucose was the starting material that we had in the beginning. By the end, we are getting in anaerobic conditions. One way we are getting lactic acid, in another way we are getting ethanol. What happened here? With the change in environment, with the change in the substrate, with a change in the enzymes, what will happen? We can get a different byproduct, okay? So in a biochemical reaction. So that is the nice part about it, okay? So what happens in enzymes? What will happen? Using the same starting material, we can get two different byproducts with a change in the environment, with a change in the in the enzymes, okay, and in the organisms also. Understood? So that also diversity exists in enzymes. Okay. Another property: how does enzyme participate in a reaction? I told you, in a chemical reaction we have a starting material which we call it as a substrate. An enzyme has to react with a substrate. Understood? So how does this reaction happens? Why does enzyme specifically specifically recognize a substrate? See, if you talk about a biological system, in simple terms, you can tell that there are thousands of chemical reactions spontaneously happening. It's a huge biochemical factory. You won't believe the amount of chemical reactions happens. Okay, it is so many. So when this is happening, is there a possibility that a particular enzyme can misrecognize a substrate? Okay, MSL, right? Misrecognize a substrate and then give us some other product, let's say fake product. Does it give us fake product? Is it possible? It is not possible at all. Why? Because enzymes being proteins and proteins being so structurally specific. Now, what happens is enzymes recognize a specific regions of the substrate and they are highly narrow to the specific recognition. So that means they pick their substrate very specifically. They don't have multiple what do you call substrate. So when they recognize this specific substrate, they go and bound at the active sites of the substrate. So when they go and bind to this active sites of the substrate, now what happens is there is a complex formation which we call it as enzyme substrate complex. So when it forms this enzyme substrate complex, it is just like a very interesting story. Okay, to divert a little bit, how many of you have seen this Interstellar movie? Okay, have you watched this Interstellar movie? Just give a thumbs up or no. Interstellar? No. Yes, you watched. Excellent. Right. So, anyways, if you have watched this movie, it's a very relative thing to understand. You know that at one point, these people are in a one ship and they have to go and attach to the main ship and they perform this docking while the spaceship is spinning. So this small thing goes and attaches to the dock and the dock locks. Right? Do you remember that scene? Like even in International Space Station, the same thing happens. What will happen? We send a small satellite, and the small satellite becomes a bigger part of International Space Station, right? So what is happening here? It goes and attaches to this specific point called as a dock. So in dock, what happens? It goes there where the doors are there for it to come and attach. So it goes, clamps like this. Can it attach to any other point there? No, it's not possible. Why? It's simply not designed for that. Exactly the same thing happens even in enzyme and substrate interaction. In enzyme and substrate interaction, what is happening here? Enzyme recognizes a specific dock, and which we call it as an active site, and they are manufactured in such a way that the enzymes are manufactured in such a way that it goes and locks very specifically at this three-dimensional keyhole, I can tell. So it goes and sits there, okay, to forms this complex called as enzyme substrate complex. Why do we have to produce this enzyme substrate complex? Now, to any chemical reaction that we are talking about, let's say that a substrate is converted to a product. So during this chemical reaction, what will happen? Let us say that we require a tremendous amount of energy to convert this substrate into a product. Now, what happens is, sorry, I will write it a little bit clearly. See, let us say that we have a chemical reaction where it requires this much amount of energy to get a product. Okay, so this is the amount of energy. This is the energy. This is the time. Okay, so this is the amount of energy we require. Now, if I use an enzyme, what will happen is a substrate combines with an enzyme to produce a transition state or a complex state. Now, since this is a protein and a on an organic molecule interaction, now what will happen? The structural configuration changes. When the structure changes, the amount of, see, for example, just by rearrangement of bonds, you can save so much of energy, right? So instead of directly breaking some bond, you can transit into some other bonds. So thereby, for example, breaking a covalent bond requires higher energy than breaking an ionic bond in simple terms. Similarly, when an enzyme and a substrate interacts, now the chemical configuration changes, their structure changes. So that will create this new product, or a complex which is called as enzyme substrate complex. So this transition state will ensure that a very little amount of energy is required to get a product. Okay, so now what will happen? This enzyme substrate complex, when it is formed, it forms this transition state. Once this transition state is achieved, okay, now what will happen is the enzyme will fly away, you get a product, and then you have your enzyme and like you have your what do you call substrate coming up. See, here it is very simple that you have to understand that the interaction of a substrate with an enzyme happens very specifically, just like you have a key and a lock. So that happens because you need to produce this transition state, and transition states reduce this energy requirement. Once that is happened, now you get your product and you get your enzyme that is separated out of your product. As simple as that. Okay. See, when this is happening, let's say, let me tell you one more thing. So while the enzyme and substrate interacted, correct, right? And they form this complex, and this complex is leading to the product formation. Now, as the product is being formed, now, see, substrate is converted to this product, right? So as it is converted to this product, now what will happen is the activation site for this enzyme that was there initially, it will become completely it's not having that affinity. Affinity means it cannot go and recognize it anymore. So when it cannot recognize it anymore, now what will happen is the enzyme will disassociate, okay, leaving the product alone. Simple. When it was in a substrate form, enzyme could recognize it, go and lock into it. When it became a product, the same site is no more recognizable. Why it is no more recognizable? The structure, the composition, everything changed. Now the enzyme can no more recognize it. When it can no more recognize it, now the enzyme will come out of it and the product is released. Okay, this is because the affinity between this enzyme and product is very less compared to that of enzyme and substrate. Got it? Did you understand this, students? Yes. Okay. Others? Are you following? Okay. So if you have any doubts, please leave it in the chat box. All right. So let's go back to where we saw. Okay. Now, while we were talking about how an enzyme goes and recognizes specific regions in the substrate, I told you that when this transition state is happening, right? Somewhere, somehow, the energy demand to complete that chemical reaction has decreased tremendously, correct, right? So this was the initial talk or initial discussion. I told you that this is the amount of energy I require to complete a reaction. But when I put an enzyme, okay, now what happened? Suddenly, the energy requirement was so low and the time was also very fast. How did this happen? So for that, we have to understand the concept of activation energy. So how does this energy decrease happens? Okay. So if we talk about any chemical reaction, now what will happen in a chemical reaction? You have a substrate, and to this substrate, you require certain amount of energy, okay, to get a product. Why do you need a certain amount of energy to get a product? Why? Because see, for any chemical change, the change requires the application of energy, as I told you, to make bonds or to break bonds. So this process has to happen. So if we take like that, when you have a substrate and a substrate is becoming a product, now it goes into this transition state. Let me talk about this transition state in the form that you understand more. Okay. Now we have, let us take a metal, and we are heating a metal. When we are heating a metal, the metal becomes a soft, right? So it is losing its solid state. It was very hard. and now it is becoming softer. The walls are becoming softer, where if you just try to push it hard, it becomes a dent, correct, right? So this state where it is almost molten before it is going to become liquid, so this state is called as a transition state. To achieve this state or the middle state, you require some amount of energy. What is the energy that you put? You put a lot of heat energy to convert this iron block into this molten block, solid iron block into this molten block. What was your aim? Your aim was to melt this iron. To melt this iron, is it happening in a suddenly? No, it's not happening. So what is happening? You need to put more energy. So when you put more energy, now automatically this iron soften and it become a transition matter, like a plasma, or right? So or molten. So this molten becomes liquid. So this time where it takes to reach the middle state of a chemical reaction is called as a transition state. So now what happens? For every chemical reaction, we need some amount of energy input, and that energy input we call it as an activation energy. See, activation energy, by definition, what it says, it is the energy required, okay, to start a chemical reaction. So that is called as an activation energy. Now, if you take any chemical reaction, so this reaction, either it will be an exothermic reaction or an endothermic reaction. Now, how do we call it as an exothermic reaction or an endothermic reaction? Now, whenever a matter or a chemical reaction goes through a higher energy state. See, for example, to convert this solid iron, we had to increase the energy, and then it became molten, and suddenly it became, what do you tell? You need a lesser and lesser temperature to convert this into a liquid, correct, right? So now you applied some amount of energy to reach the higher energy state. So that is why the molecules vibrated and then they started becoming molten. So this external input of higher energy is called as a transition state energy. So whenever that happens, now what you get? You get an exothermic reaction. What I was saying, whenever we have a chemical reaction, in this chemical reaction, we need to put additional energy to make this chemical reaction to go from a normal lower energy state to the higher energy state. So when we take this substrate from lower energy state to the higher energy state, so that is called as a transition state energy. Understood? So what does our transition state gives? Our transition state gives a transition molecule, correct, right? Or an intermediate molecule. That is it. Now what will happen? So this activation energy, what we understood, right? What is this activation energy? If you take the difference between the average energy of the substrate and the transition energy, what is this average energy? For example, so this is the flat line where you get from substrate to product, correct, right? Okay, this is the product. Now this is the substrate energy. Now from the substrate, if you have the transition state to which it has reached, so the energy that was required, okay, to achieve this transition energy is called as an activation energy, correct, right? So we had the substrate, we had to input some energy to take this substrate to the higher transition energy. So that difference is called as activation energy. Now listen to this very carefully. Whenever we have a product, okay, so that is in the lower energy, and then you have a substrate, okay, and this difference is there, right? You take a product and you take a substrate. Now when you compare the energies levels of this substrate and product, and if the product is in lower energy, okay, then it is always an exothermic reaction. Understood? So in an exothermic reaction, what will happen? You require no energy, okay, to form a product. Understood? Whereas if you take it in the ulta, like if a higher energy level is there in the product side compared to that of a substrate side, now it will always be an endothermic reaction. So remember this. This is a very important point that you have to remember with respect to your NEET examination. So they have asked, if the product is at a lower energy than the substrate, okay, if the substrate has to be in the higher energy, right? No, substrate will be in the lower energy than the product. So what happens here? What kind of a reaction can we expect? We expect an exothermic reaction, or it is also called as spontaneous reaction. So if this statement comes, your option should go for exothermic reaction. Understood? So now let us talk about another aspect of the energy, that is in a biochemical reaction. So whenever we talk about a biochemical reaction and the energy requirements, what does enzyme play a role? What kind of role enzyme plays? Enzyme, since it can go and bind to the substrate and change its chemical nature so that it can easily achieve this transition state. Okay, always enzyme reduces the amount of activation energy that is required. See, without the enzyme, the activation energy is very high. Correct, right? We need more energy to get the product. Whereas enzyme, what it does is it reduces this requirement of the higher energy. So that is why with the presence of an enzyme, you have low activation energy. That is why our speed of reaction and the rate of reaction or the velocity of the reaction increases. Understood? So next topic that we have to discuss about enzymes is factors that is affecting the enzymes. Okay, did you guys understand? One second. Yeah, students, did you understand or should I repeat? 39. Okay, thank you. So let's get back to the activation energy. Oh, sorry, factors affecting the enzymes. So when we have the factors affecting the enzyme activity depends on three major factors. Temperature and pH is one common factor. Second one is concentration of the substrate and presence of an inhibitor. Okay, so these are the three things, and we will discuss about this in a little bit of details. So what is this? What is the meaning of factors affecting enzymatic activity? Let us say that we take an example of a human body. In human body, we always maintain this temperature, body temperature of 38 degrees Celsius, normal human body temperature. Why all of these warm-blooded organisms like human beings, they maintain this constant temperature irrespective of whether our climate is getting cold or it's getting very hot. The reason you tell that it is because to maintain this balance called as homeostasis. So one of the reasons why your body needs this temperature balance is because many of the enzymatic activities is temperature sensitive. Okay, since enzymes are proteins, now this activity of the proteins depends upon what temperature they, what temperature they act in. Correct, right? So whenever there is a higher temperature, you take a protein and you keep on increasing the temperature. What happens is, because in higher temperatures, the peptide bonds between these amino acids can break and it can degenerate the structure of the protein. When the protein structure gets distorted, now the protein will not become of any use because it can no more recognize this active sites on the substrate. Okay, this is called as denatured proteins. Similarly, the same thing happens with respect to lower temperature. Whenever you try to, you might have heard about this new experiments where people are trying to freeze themselves, okay, before instead of dying, so that they believe that in future that their frozen dead bodies can be given life to, which is called as cryopreservation, right? We do this in with respect to seeds and pollen grains and everything. Whereas talking about live tissues or live organs, or when you talk about human bodies, okay, things are happening. So when you talk about these things, what is happening? Why don't the same thing happens when we burn a body? When we burn a body, what happens? Everything will become turned to ash. Why did it happen? Whereas when we freeze a body, the same thing is not happening. Why? Because at higher temperature, you know that the bonds get destroyed, and then that will lead to denaturation. Whereas in lower temperature, what happens is it freezes. Since it freezes, the structure of this enzyme remains intact, whereas nothing changes. When you defreeze it, the same enzyme can participate in a chemical reaction. Whereas the enzyme that is given temperature, that at higher temperature, so at higher temperature, since it degenerates, okay, it will be of no use when you bring back the temperature down, okay, in many of the chemical reactions. So that is why enzymatic activities are highly temperature specific. So is there a specific temperature in which an enzyme participates? Yes. So this temperature at which we can have a highest enzymatic activity, where we can find this highest enzymatic activity. So that is called as an optimum temperature. So for example, we have this temperature requirement and we have this enzyme activity. So here in temperature, I can write it as 10 degrees, 20 degrees, 30 degrees, right? And 40 degree, like as I move on, it can go on increasing. So enzymatic activity depends on how much of product we get, correct, no people? So here enzymatic activity is determined by, for example, if I have 100 enzymes, okay, and then I have 1000 substrate, okay, all these 100 enzymes should participate with the substrate to give me product. Simple. So this participation, all of them are participating and all of them are giving highest productivity. So this happens where? At the optimum levels, correct, no people? So this temperature at which where we can find the highest participation of the enzyme, this is called as optimum temperatures. Here we get the highest product also. Understood? And as the temperature decreases or increases, now this optimum level will shift. Uh, Always when you shift towards or against this optimum level, now what will happen is the enzymatic activity changes. As we go towards this, okay, see, we are moving in this direction, right? As I move towards this optimum temperature, I get more and more product. And as I move away from this optimum temperature, what is happening? The enzymatic keep on decreasing. Okay, so these are the very important factor. The same thing happens with pH. One more example I would like to give about temperature. So you can understand that most of our warm-blooded organisms and higher organisms, they participate at a very fixed temperature, okay, by maintaining this balance, okay, homeostasis, okay, which we call it as warm-blooded organisms. Whereas we have certain organisms in the lower lower kingdoms, such as you have monera, where you have archibacteria called as thermophilus uh, bacteria. Okay, so this particular bacteria can live in a very high temperature in hot thermal vents. For example, you have your hot springs, right? So where the water comes out from the ground. So here the temperature is almost up to 80 to 90 degrees Celsius, and even the pH is around 1 to 2. Very acidic pH and very high temperatures. So how are these bacteria surviving? The bacteria make use of special enzymes which have the capacity to survive in certain temperatures, in that high temperatures. This is the same thing does not happen in many of the higher organisms. Why? Because it leads to degeneration of proteins, okay, at higher temperatures. Okay, so this is about being temperature and pH specific. One more example for pH is you can take an example of your stomach and you can take an example of your small intestine. The enzymes that participate in the stomach require acidic pH, whereas the enzymes that participate in your small intestine, many of them, they depend on basic or alkaline pH. Okay, so it's quite opposite pH. Here also they're pH specific. The small intestine enzymes cannot become active in the stomach. Okay, the same thing, the stomach enzymes okay, cannot have higher effects uh, in the small intestine. So this is the temperature and pH specificity with respect to enzymatic activity. Understood? Okay. The next factor is actually substrate concentration, but students, we are running out of time. Okay, we'll talk about it tomorrow. This is a very important topic. I hope you understood. If you have any topic that you need to or you need me to discuss again, please let me know. Okay, in the comment section, you have 3 minutes.